Intestinal glutamine. So, what is glutamine? Glutamine is the non-essential amino acid and most abundant amino acid in human blood and skeletal muscle, which play a role as energy intermediary and a substrate for the synthesis of peptides and non-peptides, while also contributes to the detoxification of ammonia and acid-base balance. Although normally considered non-essential, glutamine becomes conditionally essential during catabolic state as proven by observation. Facts: 30% of total glutamine utilizes by intestine, which indicate it is a key nutrient for intestine. Source of glutamine can be obtained from endogenous sources, which are produced within the body, where glutamine is synthesized in skeletal muscle, lung, adipose tissue, and brain. In addition, glutamine can also be obtained from exogenous sources, which include dietary protein intake and supplementation. Next, the role of glutamine. Firstly, tissue integrity. Glutamine support rapid epithelial renewal, helps maintain intestinal barrier integrity via tight junctions, and reduces intestinal permeability and protect barrier function. Secondly, apoptosis and cellular stress. Glutamine helps maintain balance between proliferation by preventing ATP and apoptosis, exhibit anti-apoptotic effect, and enhance SSP expression. Thirdly, inflammatory pathway. Glutamine suppresses intestinal inflammation, inhibit NF-kappa B signaling pathway, and modulate ST-ATP signaling pathway.
For initial protein breakdown, dietary protein containing glutamine broken into smaller peptide by the enzyme pepsin. This prepares the protein for further digestion in the small intestine. Glutamine enters the enterocyte from the intestinal lumen via the B0 transporter. According to the Holyset 2024, from the bloodstream, glutamine enters via the SN81 and SN82.
The enterocytes use glutamine as their main energy source instead of glucose. During stress or injury, glutamine uptake increases to support cell growth and repair.
In the enterocyte, specifically mitochondria, glutamine rapidly hydrolyzed by water into glutamine and ammonia by glutaminase enzyme.
This is the first regulatory step linking glutamine to energy and nitrogen metabolism to control nitrogen release and increase the rate during metabolism.
After that, the amino group from glutamate transferred to oxaloacetate by aspartate aminotransferase, forming aspartate and alpha-ketoglutarate without releasing free ammonia. Alpha-ketoglutarate enters the Krebs cycle and used to produce energy for the enterocyte and also in ADH and FADH2, which then donate to the electron transport chain to generate another ATP. However, not all glutamine is used to produce energy. Some of it is used to make a glucose, especially during fasting state. Through the Krebs cycle, alpha-ketoglutarate also used to release an oxaloacetate, which is important for making glucose. Oxaloacetate is converted to phosphoenol pyruvate by the enzyme pepsicase. Then it is used in several steps to form glucose. This intestinal gluconeogenesis helps keep blood glucose stable during fasting or after meal. Other than that, oxaloacetate that form phosphoenol pyruvate are converted into pyruvate by pyruvate kinase. This pyruvate acts as a substrate for the formation of alanine through transamination with glutamine, catalyzed by alanine aminotransferase. Through reduction reaction, pyruvate are reduced by lactate dehydrogenase to produce lactate. Through oxidation reaction, NADH is oxidized back to NAD. Once alanine forms, it leaves the cell through a special transporter on the basolateral side, which mainly LAT2. For lactate, because it is a charged molecule, it needs a special transporter called as monocarboxylate transporter 1, MCT1, which moves the lactate out together with the hydrogen ion.
In the liver, alanine and lactate serve as important substrates for gluconeogenesis, allowing the glucose to be produced and released into the bloodstream. This process helps maintain normal blood glucose level, especially during fasting, illness, and physiological stress.
Glutamate also contributes to the synthesis of ornithine. Ornithine reacts with carbamyl phosphate to produce citrulline as part of urea cycle. Other than that, glutamate also produces other amino acids such as proline and arginine.
The ammonia that is released from the glutamine earlier is toxic and must be removed from the body. The ammonia that is released will enter the bloodstream and will be transported to the liver. Inside the liver cells, ammonia first enters the mitochondria where it is converted into citrulline. Citrulline then moves out of the mitochondria into the cytosol. In the cytosol, citrulline is further processed to form urea, which is a non-toxic waste product. the urea produced is released into the bloodstream and transported to the kidneys and finally excreted from the body in urine. Through this pathway, known as the urea cycle, the liver will safely remove toxic ammonia and maintains the nitrogen balance in the body. Glutamine is also a major nitrogen donor in enterocyte. It donates nitrogen to purines, pyrimidines, and non-essential amino acids. It emites nitrogen to support synthesis of nucleotides and non-essential amino acids that is required for rapid cell division. This process helps to support the high enterocyte turnover and the DNA and RNA synthesis.